The start of the Glasgow Marathon. An annual challenge for people in the peak of condition to test their endurance. But it's also a major fundraising event for medical research. Over 400 of these runners are being sponsored for research into muscular dystrophy, a hereditary disease that causes wasting of the muscles. The money they raise will go to help find a cure for boys like Lee and Damien Evans, who both suffer from the most common and most severe form of muscular dystrophy called Duchenne, after the French physician Guillaume Duchenne, who first described it over a century ago. Before the age of 3, boys like these begin to lose the use of their muscles. It starts in their legs, and before adolescence they lose the ability to walk and become wheelchair-bound. This gradual weakening of the muscles eventually spreads to every part of their body, and it's untreatable. In the majority of cases, Duchenne muscular dystrophy is inherited by a boy receiving a defective gene from his mother. But no one knows which gene is the culprit and why it wreaks such havoc in the body. Until the Duchenne gene is located and we can find out what it normally produces that is so essential to the working of healthy muscles, then the cause of this disease will remain unknown. But what was once a scientific daydream is fast becoming reality. The mapping of genes on chromosomes is being made possible by new methods of manipulating DNA, the chemical substance from which all genes are made. In different laboratories, they've been trying to isolate and sequence the Duchenne gene. In Toronto, Dr. Ron Wharton. In Oxford, Dr. Kay Davis. And in Boston, Dr. Lewis Kunkel. Their task is a marathon of another kind. But in this case, it's not who wins that's important. It's that the race is won. The scientists are spurred on by the need to solve what is a common human problem. Duchenne muscular dystrophy affects all races, but usually only boys.
All right, Dion, I'd like you to try to stand up for me.
1 in every 4,000 boys are born with it every year. Because the disease is sex-linked, we know that the gene has to be somewhere on one of the sex chromosomes.
Very good. Very good. Okay.
But the discovery that gave scientists their first breakthrough came a few years ago from labs like those of Dr. Ron Wharton in Toronto, where they were studying the case of a girl who showed classic symptoms of Duchenne muscular dystrophy. Such cases were rare and unexpected, as girls don't usually suffer from the disease. It was examination of the girl's chromosomes that provided one of the vital clues to the location of the gene. If chromosomes are stained, they show a distinct pattern of banding under a microscope. Each of us carries 23 pairs, and we inherit one member of a pair from each parent. The pair of sex chromosomes determines whether we're male or female. A boy has 1 X chromosome from his mother and a smaller Y chromosome from his father. The Duchenne gene is carried somewhere on the X chromosome, and if a boy inherits a defective gene, then he will get Duchenne dystrophy. Girls have 2 X chromosomes, and if they carry a defective Duchenne gene on one X, this is usually compensated for by a normal gene on the other X. For a girl to suffer from the disease, she would be expected to carry a defective gene on both X chromosomes. But in Ron Wharton's patient, the cause was different. It was due to an irregularity of one of her X chromosomes. Attention focused on a particular band on the short arm of the X chromosome. for it was in this region that one of her X's had broken. The broken end had become exchanged with part of another chromosome. Such an event is called a translocation. One consequence of the exchange was that the normal X chromosome became suppressed. Only the translocated X was active. So this chromosome was responsible for the expression of the disease. Could it be that the break had disrupted a normal Duchenne gene? That was the conclusion they came to at the Toronto laboratory. Dr. Ron Wharton.
I think the important thing about the girls with translocations is that there were a number of them. By 1981-82, there were about 5 or 6. And the crucial point was that in every case, there was a rearrangement of the X chromosome. And in every case, the rearrangement took place at the same site in the X. And we concluded, and others concluded as well, that that had to be the site of the Duchenne gene, because all the patients had their chromosomes broken at the same place. What do you have this morning?
Although it was clear that the breakpoint on the X chromosome lay in or close to the gene, isolating DNA from that region would prove elusive for some time yet. DNA can be isolated from cells, but even under the most powerful electron microscope, it shows up as a tangled mass. To pick out a single gene, you would need to take one of these tiny clumps and unravel it. In a chromosome, coils of DNA are wound around lumps of protein. Take away the protein, and you'll see that DNA is like a twisted chemical ladder. There are 2 strands held together by rungs of 4 different chemicals called bases. The order in which bases are arranged is crucial, because the instructions for making the proteins needed for life are written in this 4-letter alphabet: A for adenine, C for cytosine, G for guanine, and T for thymine. Over 300,000 bases may make up a single gene, still a minute amount of the total DNA in a cell. The task of finding something as small as a gene is like looking for a footprint on a 26-mile marathon. But scientists knew they had to look along a particular stretch. One X chromosome translates to about 1 mile of a marathon. Now they had their first landmark on the route, the breakpoint on the X chromosome in girls suffering from Duchenne. But was this site also important in boys with the disease? The answer to this question was provided by Dr. Kay Davies, working at the University of Oxford. She decided to look for stretches of DNA as markers on the X chromosome that were close to the suspected site of the gene.
Our first objective was to purify the X chromosome so that we could isolate sequences from it which were on the short arm. Now, if Duchenne muscular dystrophy really was on the short arm, then we could find DNA markers close to it, and they would be co-inherited with the Duchenne muscular dystrophy gene. So if we take 2 markers on either side of where we suppose it is, then whenever we got co-inheritance of this, we should get co-inheritance of the Duchenne muscular dystrophy mutation.
Her search for markers began by making an X chromosome library. The X chromosome can be chopped into bits and grown up or cloned in bacteria, so that each bacterium contains a different piece of the X chromosome. To find out which bits might carry muscle genes, she first looked to see which genes were active in muscle tissue. She extracted and radioactively labeled muscle genes and added them to the culture of bacteria containing bits of the X chromosome. In this way, she was able to screen muscle genes across the X chromosome library in search of muscle-coding sequences that mapped on the short arm of the X chromosome. Any bacteria whose DNA matched up with muscle genes would be marked out by areas of radioactivity. By exposing the plate of bacteria to photographic film, those bacteria containing the bits of X chromosome that she was interested in showed up as black dots. She could isolate the X chromosome DNA by picking the bacteria in this part of the plate. Kay Davis succeeded in isolating DNA markers that flanked the site of the breakpoint in girls with translocations. These markers were co-inherited with Duchenne in boys. So here was further evidence that confirmed the gene's location to be somewhere in the vicinity of the breakpoint. Such markers could be used as DNA probes to trace the inheritance of the faulty gene. So there was an important spin-off from her research for the families of children with Duchenne. An accurate way of detecting carriers is particularly important for women. 2/3 of all Duchenne cases are inherited on the X chromosome that boys receive from their mother. The same chromosome could also be carried by other female relatives, and if so, could be inherited by their children. A mother usually has no idea that she's a carrier unless she has a son with the disease. Even then, it's not certain whether she has passed on the faulty gene. It could have arisen by a mutation in her son. In Mrs. Evans's family, there had been no previously known history of Duchenne muscular dystrophy until her first son, Lee, began to show symptoms.
Yeah, boys, how's it going? Alright. Who's winning? Miracle. Cheating.
Well, Lee had been falling over and couldn't walk very far for quite a little while. So we had an appointment to go up to Guy's, and he had various tests. And the doctor told us that Lee had a disease called muscular dystrophy. We didn't know anything about muscular dystrophy, what it would do to him, what it was doing to him. We didn't know anything at all. But he told us then that there was no cure. We didn't have any idea that Damien also had muscular dystrophy until a few months later when Dr. Guy's asked us to see him again, and he suggested that Damien had some blood tests.
Who's was that, Lee? Come on in, James, you're in the lead.
We just didn't realise that muscular dystrophy could affect Damien as well. We thought that it was just something that had happened to Lee, but unfortunately it wasn't, because Damien had some blood tests done, and the doctor called us again, and he told us that what they had found in Damien's blood and Lee's blood was the same, and that after various tests that Damien also was a sufferer of Duchenne muscular dystrophy.
Before DNA probes were available, only a limited analysis could be made of the rest of the family. But now, several gene probes, similar to those discovered by Dr. Kay Davies, can be used to try to identify the affected X chromosome. Professor Martin Bobrow at Guy's Hospital uses probes to predict more accurately what the chances are of a woman being a carrier of Duchenne.
Now, since you have two sons, both affected with muscular dystrophy, it's very likely, almost certain, that you're a carrier of the abnormal gene yourself. Yeah. This is you on the pedigree chart. These are your two sons, Lee and Damien, your mum, your three brothers, and your sister. In your family here, your brothers have been perfectly normal. They haven't had muscular dystrophy. They may well not be perfectly normal. You know that better than I do, but muscular dystrophy they've not got. That would have made me think beforehand that your mother might well not have been a carrier, because she'd be quite lucky, if she is a carrier, to have three normal sons.
Yeah.
As far as your sister is concerned and the chances of her being a carrier, the question that we've got to answer before we can really start to look at that is whether you inherited this muscular dystrophy gene from your mother, or whether it's something that just started off in this part of the family for the first time, because we don't know of any other family history further back of muscular dystrophy. So we've taken some blood samples from both your mother and yourself for this muscle enzyme test. We know that the levels of this muscle enzyme, when measured in the blood, are unusually high in at least some people who are carriers of muscular dystrophy. It's not a very accurate test, but it's quite a good guide. What we've found is that, not surprisingly, the levels in your own blood are high, and that confirms that you're very likely to be a carrier,
Yeah.
that your mother also has quite high levels of muscle enzyme in her blood. So you have probably inherited this from your mother.
Yeah.
If your mother has the gene as well, we'll mark her in as a carrier, then there must be quite a significant risk of this having gone through to your sister,
Yeah.
and she could, in principle, pass it on to other children of hers. In order to try and clear up what's happening, we therefore ought to go on and do these new DNA tests on blood samples from the whole family so that we can try to trace which chromosome in your family has got the muscular dystrophy mutation on it, where it's come from, and whether it's got into your system or not.
DNA is extracted from white blood cells in each of the samples. These small threads are from 10 million cells or more. The DNA is broken up into smaller, more manageable fragments by adding a substance called a restriction enzyme. This enzyme acts like scissors, cutting the DNA at specific points. Once digested, the fragments are mixed with a fluorescent dye, and samples from different individuals are loaded separately into wells on a gel. An electric current applied across the gel creates a gradient down which the fragments will migrate and separate out according to size. Suppose this is a piece of DNA from one member of the family, with its sequence written with the 4 bases. The enzyme always cuts the chain at a particular sequence of bases, which it recognizes. So wherever this sequence occurs, the DNA will be cut, and there'll be a lot of fragments of different lengths. Electrophoresis separates the fragments according to size. The pattern is a genetic fingerprint. This person has a pattern of DNA fragments with one size of 10,000 bases, 2 14s, 3 around 16, and so on. But in some people, there might be a misprint, and then the enzyme will not cut at this site. So the pattern of fragments differs according to the number of sites recognized. For example, there might be no 10-unit segment and one less 20, but there might be one 30 units long. To check whether the enzyme has done its job, the gel is examined under ultraviolet light. About 100,000 bands of DNA are clumped together in each lane of the gel. To pick out bands from the X chromosome near to the gene, an imprint of the fragments is transferred onto filter paper and then analysed by adding radioactive probes. Fragments of DNA that match any of the probes will show up as dark bands on a photographic negative of the gel. By comparing the results for Lee's X chromosome with the rest of the family, Professor Bob Rowe will be able to see whether anyone else is carrying the same affected chromosome.
This is Mrs. Evans herself, showing a homozygous pattern. This is her normal brother, and this is her sister, who also shows a heterozygote pattern.
How many other probes have you done on this family?
We've tested them with 6 probes in total,
Yes.
and the only 3 informative ones, i.e. we can distinguish chromosomes, are the 3 shown on the pedigree.
And we therefore presume that it is this chromosome here with the 8.7 band that carries the Duchenne mutation.
Yes, that's what we think.
Right. Hazel, we've redrawn the pedigree chart and tried to show on it this time the results of all the DNA probe tests that we've done on the blood samples that we've had from you and from the members of your family. We have all the results of the DNA tests drawn in bright colours so that we can follow easily where the different chromosomes have come from. Your boy has a chromosome with these 3 markers on, and we can tell your 2 chromosomes apart. We can see the pink one that went to your son, and we can see that that pink one with the 4.4 and the 8.7 and the 22 on it is one that came from your mum. That's what we would expect, because we think that she is a carrier of dystrophy, that she has passed that to you on this pink chromosome, and that you in turn passed it on to your son. Your mum's 2 chromosomes are different from one another at this point and at that point, 4.42.2 and 8.77.5. We can see that the yellow chromosome, the other one, the one that you didn't get, has been passed to your brother, and the fact that he is normal makes sense, because it says that she has 2 chromosomes, a normal one and a Duchenne one, and that you got the Duchenne chromosome, your brother got the non-Duchenne chromosome. Your sister, if we look at her 2 chromosomes, we can see that the pattern of the green one comes from her father. She must have her father's chromosome. And she has also, from your mum, received that yellow chromosome, the same one as your brother, not the same one as yourself. So she is very unlikely to be a carrier. There is a chance of error, as you know, because the chromosomes can mix themselves up, they can swap pieces, but it's really quite unlikely, and particularly since we now know that her muscle enzyme levels are rather low as well, the chances of her being a carrier are really very small at this stage. Now, as far as you're concerned, we can tell these 2, the pink and the green chromosome, apart, and if you were to have more of a family, we could do that on a sample taken during early pregnancy as well. We could do the DNA tests on a sample from the pregnancy and tell not only whether it was a boy or a girl that you were expecting, but whether, if it was a boy, whether it was a boy with the Duchenne mutation carrying chromosome or the other chromosome. And we could say quite accurately, not absolutely, but quite accurately, whether that boy would be likely to have the disease or not.
So the Evans family now know where the gene has come from, who is a carrier, and they could find out the likelihood of any future children being affected. But how successful is analysis with probes in other families?
That depends firstly on having a sufficient number of other people in the family. There must be both normal and affected boys or men in the family so that one can tell what's going on. And secondly, there's an element of luck. It's a question of whether the women who are looking for advice have the right combination of probes so that we can clearly tell their 2 chromosomes apart from one another. That happens at the moment in about 95% of families with the range of probes that we are using now, and that will improve, but there still are occasional situations where you can't do it.
Although helping families can reduce the number of boys who will inherit Duchenne in the future, as yet, research has made no impact on the condition of those already suffering from the disease.
Walking has got very hard for both the boys, especially Damien, but we push them on and we try to get them to walk as far as possible because we don't want them to just give up and then go into the wheelchair straight away. So we take the boys swimming because the physio has explained to us that if the boys can learn to walk in the water, it will help them walking about at home or at school or wherever. So we went up to Guy's and physio at the pool with both the boys and gave them confidence in the water, and also because it's a sort of activity that their friends can do as well when they're not cut out from. They can also go swimming the same as their pals can.
A way of halting the relentless progression of Duchenne dystrophy must be found, for the disease can strike any family for the first time. Identifying carriers can never completely eliminate the problem.
At least 1/3 of the cases of muscular dystrophy arise as a result of a fresh mutation in each generation. So they're born to girls who are not carriers. And even if you could detect every carrier in the country by testing all schoolgirls or something of that sort, you would still be left with that 1/3 of cases that are arising as spontaneous mutations, which you'd not be able to get at. And having some means of treatment would seem to me a very important part of one's long-term objectives.
Okay, we're going to have a race now, see who gets first back to mummy.
The biggest single thing that we're hoping for is that we will go from just tracking chromosomes to actually knowing what the mutations are in most patients. If we knew what the gene actually made, and if we could test for that, then we wouldn't have to just infer whether the bad or the good chromosome had got into a particular person. We could actually test on a yes-no basis whether they were or were not affected, were or were not carriers. And then, looking further ahead than that, if we knew what the gene product was and what its function was, we might have some sort of opening towards actually being able to treat affected people, and that would be the biggest prize of all.
All right.
Hazel's friends and family do all they can by raising money for research.
When we found out that the kids had muscular dystrophy, we felt that we needed to be doing something. We've got to make the best of the situation as it is.
These are the ones that we've got finished.
So we try and get as much fun out of fundraising for us, for the kids. We try and have fun and a laugh. We get the children very much involved. The kids absolutely love a jumble, but especially the Christmas one. I think the kids are more excited over helping in the jumble than what they are at Christmas at the moment. They all come, they all do their little bit, and we aim to make a lot of money today.
Come on, Christmas crackers, $1.25 a box. Come on.
Nah, don't like no. I bought you some bits, Brenda.
10p a ticket and 6 for 50. Come and help our boys. Help research.
94% of all the money paid in is all spent on research. And that makes me feel that people that support us in anything that we do for research, their money is going to the cause that they want it to go to. The Muscular Dystrophy Group has a lot of branches all over the country, and all those branches make lots of money. The South East London branch always back us up. They always help us if we're short-handed or we want ideas.
So far this year, with the help of the publicans who did the Interclub and the marathon runners and marathon tickets that we sold, we've done 13,500. And I reckon we'll have 15,000 before the end of the year.
Became involved. I had a son. He died in 1956 at the age of 18. And it was from there that I feel, you know, I'm a founder member of Muscular Dystrophy. And still 30 years on, I'm still here.
This is what I can do. I can make money for the group for research. It gives me the hope for the future, because without the fundraising, we've got nothing. With the fundraising, we've got a lot of hope, and we are getting there. I know we're getting there.
From the head office to the littlest committee, we're all there for one thing, for muscular dystrophy, to find the answer. That's all our prayers are for. Please God, we'll be soon.
In the United States, the Muscular Dystrophy Association is by far the biggest fundraising organisation, and they've successfully enlisted public support with the help of top show business entertainers.
Ladies and gentlemen, Mr. Jerry Lewis.
Every August, on an American holiday, Labor Day weekend, Jerry Lewis hosts the annual telethon, a round-the-clock television appeal broadcast all over North America.
Thank you, ladies and gentlemen. We're here to do some important work while at the same time trying desperately to entertain and to make it as light as we possibly can, knowing all along there's nothing light about it. We try to bring you the best possible show we can for the next 22 and a half hours. And we try to do that with dignity, with respect, and with a deep concern for the reason that we're here.
Hi, how are you? Hi, so you have your braces on?
For the past 37 years, Jerry has been a key figure in the MDA's fundraising campaign. During that time, he's helped to raise $900 million for research into neuromuscular diseases.
How about for on both feet or just one?
The hope is that the isolation of the Duchenne gene will lead to treatment not only of boys with the Duchenne form of the disease, but also to the treatment of people with other types of muscular dystrophy.
We got the kind of research this year that could do it. It could do it. We've got the research dollars. The more we make, the more we can spend. The more you send, the more we can help. That's the name of the game. It really is an exciting, marvelous feeling to know that all of the work is not in vain, that we have a shot at it, that we can go to the pathologists and the scientists and the doctors and the people in therapy and say, here's your grant, here's the bucks, go into the lab, now find the answers.
Advances in genetics, with the need for more sophisticated equipment and larger research teams, have called for more and more money. In the last few years, fundraisers from the US, Britain and Canada have stepped up their efforts to help scientists to make that all-important breakthrough. In October 1986, the American Muscular Dystrophy Association called a press conference to announce the news that even seasoned campaigners had never really expected to hear.
Thank you very much. Good morning. It is with a great deal of joy and emotion that I announce that a group of our scientists at Children's Hospital in Boston has discovered the gene for Duchenne muscular dystrophy. This is the most common, most cruel, and most deadly form of the disease. Dr. Louis Kunkel and his research team at Children's Hospital and all of the scientists around the world who contributed to this great achievement have our heartfelt congratulations. They've worked virtually, night and day for years to find the Duchenne gene.
Can somebody explain to me how one goes about isolating a gene?
You say, come here, get out of here.
That's a question that's really very difficult because it depends on the gene you're trying to isolate. Duchenne muscular dystrophy was an example of a gene for which we had no idea what was wrong, what the product was, and we tried to use what's called reverse genetics. We knew where it was. Now we'll go about trying to, like a needle in a haystack, pick out everything else till you finally find the one that is the gene. And that's basically what we did over the course of about 3 years.
Kunkel's lead came from studying a Duchenne patient who had part of his X chromosome missing.
The boy suffered from 3 X-linked diseases: Duchenne dystrophy, 1, chronic granulomatous disease, and retinitis pigmentosa. So we have now 3 X-linked diseases and a deletion, a loss of material. And the assumption was that that loss of material was what resulted in 3 X-linked diseases. And therefore, those 3 genes must reside within that region that's lost from that boy's DNA. And that just is the same region that both the genetic evidence with markers and the translocation data had said was the region where Duchenne was. So now what we'd done, what had happened is the disease locus had been defined to be within the confines of that lost material, the deletion area.
Morning all, how are you doing?
Morning.
Tony, more seating.
If they could isolate from normal DNA the segment that was missing in the boy, then that segment should contain part of the Duchenne gene. But how could they identify what was missing? Kunkel's solution was a clever one. He took a large amount of the boy's DNA and compared it with a small amount of normal DNA that had been enriched with X chromosomes. He cut up the paired strands of normal DNA at precise points with an enzyme and then separated them. The sample of DNA from the boy was first separated before the strands were broken at random. When he mixed the fragments together, he got pairing between any that matched each other. Some of the boy's DNA reassociated with itself. Normal DNA readily found partners in the large amount of the boy's DNA. But in both cases, because the fragments were always of different lengths, the ends of these duplexes had long single strands sticking out. However, fragments of normal DNA from the region of the X chromosome missing in the boy could only pair with their original partners. And because these fragments had been cut precisely, they formed neat pairs that could be separated from the rest by cloning them in bacteria. So he isolated a number of segments of normal DNA whose absence in the boy caused the 3 diseases. One very small segment was also missing from a number of other patients with just Duchenne dystrophy. He had isolated a small segment of the Duchenne gene. The segment mapped within the dark band on the short arm of the X chromosome, some distance away from the breakpoint. Here was the first marker for the gene itself. Using this marker as a starting point, he found stretches of DNA that overlapped with it on either side. And so he continued this technique of walking down the chromosome, extending further out in both directions. But how could he find out which parts of the stretch of DNA they had walked belonged to the Duchenne gene?
I'm wondering, I can't tell because of the...
Now, we had asked the people who were collaborating with us that they would make available to us the DNA of the patients with deletions, because we realized that the deletions would then define by what's commonly deleted in a series of boys that define regions that might be important towards the expression of this disease locus. And we were able to obtain 55 DNA samples who had been demonstrated to be deletions, and then analyze them across the entire cloned region, which at that point was 137,000 base pairs, and is now subsequently almost over 200,000 base pairs. We were able to map very precisely where these deletion breakpoints were. This is the one out here.
In a collaborative effort with more than 23 research laboratories throughout the world, they found that a significant number of Duchenne boys had complete or partial deletions along the map they'd worked out.
I wonder if we can even predict it from the distance between the exon, if there's any exon.
But not all parts of a gene are essential to its function. One way of checking whether DNA is important to the proper functioning of muscles is to find a sequence that is present not only in humans, but in other animals.
Yeah, you're actually right. He actually, I think, got like a 3. If we go from this to the mouse, we could actually prove it. And so what we did was we searched within the 200,000 base pair region, but specifically emphasizing the regions that the deletions had indicated might be important. And lo and behold, we were able to identify 2 segments which were highly conserved. That is, they were maintained their nucleotide sequence between both mouse and man, and in one case, one actually is conserved into birds. So this was a segment of DNA that presumably evolution has maintained as being important, whereas the surrounding material was not maintained at all.
And yet we should take the mouse CDNAs and put them on there, and that would directly
show it as well.
If it showed 2 bands, it showed the other one that it's right underneath.
If the conserved segment coded for a protein, then they knew that they should be able to find its message or transcript.
And if that's really there, we could show...
They looked in various tissues of the body and eventually found the transcript exactly where they expected to find it.
The transcript was seen to be present in muscle, but not in a lot of other tissues. So one would then assume that this disease was that of muscle, which we already knew, and here we see the gene being expressed in muscle. So that was the evidence that this was presumably the gene.
Am I on the right track, Dr. Kunkel, when I say that if you're going to find the gene, we found the doozy? Okay. See, I put it in terms people can understand, because if he tells you, you're going to all look at one another like you're going to have a skin rash from it, because I can't get them to talk regular. You know how scientists are.
We really were quite excited, but we knew that we had a lot of work to do still. This is basically just the first step. We need to go much, much further. And as scientists, I think we really need to do a lot more. This is the first step towards an understanding of this disorder. And I would anticipate some very, very interesting results in the next couple of years about what's really wrong in the muscle of these boys.
I can only tell you that my gratitude is only exceeded by the fact that I don't know how to express it. It emotionally touches every fiber and every nerve end of my body. I didn't very honestly believe at the very beginning that we would see this kind of progress in my lifetime. For that matter, I was told by the medical community and the scientific community that I was knocking on a dead door years ago. to see this breakthrough tells me that indeed in my lifetime I'm going to see my kids better than they are today.
But Kunkel's team had not yet sequenced the entire gene, only part of it. What effect would their results have on other labs?
By publishing this, it'll also allow the competition to catch up. In other words, we've told them where it's expressed, how big it is, and where it is in the genome, and actually have given them clones and sequence information to obtain the same sequence. And that I'm sure they will. And I think actually both of the groups being interviewed on this program, Ron Wharton and Kay Davies, both have gotten theirs independently, and they also have similar information that we do. It's just not quite as far along.
Ron, we got a longer cDNA clone from the previous.
It was only a matter of weeks before another team, led by Wharton in Toronto, independently reached a similar stage in their research.
Here's the original. So that's the longer one.
They had succeeded in isolating a different piece of the gene. It came from the breakpoint of a translocated X chromosome in a girl with Duchenne muscular dystrophy.
Now, our patient was unique in that not only did she have this break in the X chromosome at what we then assumed was the site of the gene, but the part that was broken from the X was attached to chromosome 21 at the site of another known gene that had already been isolated in Philadelphia. And what we were able to do then is to take sequences from the chromosome 21 gene, use that as a probe, to find a piece of DNA that spanned the junction. So it therefore had the chromosome 21 gene at one end and the muscular dystrophy gene, or part of it, at the other end.
And this one is one of the original set too, I guess. That's what I found.
That's a great result. Congratulations. Kunkel's approach led him to a site on the X chromosome, which let's say is here. Our own approach led us to this translocation junction, which is close but not in the same spot. We have done what's called walking along the chromosome to isolate 100,000 base pairs of the chromosome. Kunkel has done the same thing from his original clone to isolate 200,000 base pairs. We have not yet overlapped, although we know we must be close. And that, plus additional evidence, indicates to us that the gene includes all of that material that has been isolated, and then some. So that we now have reason to believe that the gene might include as much as 1 million or 2 or 3 million base pairs of the X chromosome.
So Wharton's marker was next to the translocation breakpoint. Progress from this and from Kunkel's marker have since been rapid, and now they've met and overlapped. The total distance walked so far is about 1/4 of what they estimate the total size of the gene to be. The competition to get there first no doubt provided that extra stimulus. But now that one of the largest genes ever discovered has nearly been sequenced, how do the competitors feel about entering the home straight?
I suppose the Duchenne muscular dystrophy research can be described as healthy competition, because essentially it's a race. So if anyone gets a result that's an exciting step forward, then they will work that particular result up, that particular new DNA sequence up in their own laboratories, and then collaborate a few months later.
There has been one common element, and that is find the express sequences from the gene, determine the protein structure and function, and understand the disease. We've all taken different routes to arrive at about the same point now, and from here on there will be a lot of commonality of purpose as we are all attempting to isolate the rest of the gene, its protein product, determine its function, and so on.
The different techniques used to find this gene have opened up the path to find genes that cause other inherited disorders. It's a formidable task, but mapping all the genes on every chromosome is within reach, and the excitement is sending ripples through the medical and scientific community. But how soon will it be before people suffering from Duchenne muscular dystrophy will see the benefits of this discovery?
I think this is a big question that's been asked many times, is once you have this gene, how long will it take you to understand what it does? And I think that we'll have the entire nucleotide sequence and then the predicted protein sequence of this gene, if we have no technical difficulties, probably within the next year. And we're doing it both in mouse and man. I think that's very important. One can then compare the sequence, the protein sequence between mouse and man. And anything that's maintained between mouse and man are regions of that protein that are very important in its expression and its function. So we anticipate that in the near future, basically having information about the protein.
We're forming a sort of image of the protein, you're right, but it's not possible at this point to make any predictions about the exact nature of the protein. I think large genes usually make large proteins, so one would anticipate that this will be a very large protein. And large proteins are not usually the kind that circulate in the bloodstream and act, let's say, like a simple hormone. But large proteins are usually structural components of cells. So we're building an image of a large structural component of the cell, maybe a component of the cell membrane, probably involved in muscle gene function or in muscle function of some sort.
There are lots of examples of other human genetic diseases where we've known exactly what protein is involved. For example, if something goes wrong in globin in the blood, then we know everything about globin, we know the DNA sequence, the protein sequence, and yet, even over the last 10 years, we've not really been able to come out with an effective treatment or even a cure. So if it's that type of disease, then we won't be able to make any immediate progress on being able to treat Duchenne muscular dystrophy. But of course, if it's something much more straightforward, where we can replace the missing protein, then we can hope that we can treat this disease very effectively indeed, and that's why people are getting very excited.
The hope for my kids, which is so magical for them, is also a marvelous hope for those people that have been so supportive after all these years, assuming that maybe one day they'll hear, well, we tried. But no, they're hearing that we're getting there.
This is the best bit of news that we've ever had. We've always had a lot of hope for the future with the research, but we always know that before they could find the cure, they had to find the gene to find out why it was happening and what it was doing. But now they can start looking at ways to stop the boys from getting any worse. And I think that with this big breakthrough they've got, then that can be in the very near future for us.
And that was